Alright guys, so we are moving on, and our next unit is going to be all about cell division. So we're still on the whole concept of the cell. We've covered the cell and its parts and cell transport across the membrane. We've taken a look at enzymes and how they regulate metabolic reactions within our cells, and we looked at two major metabolic pathways. We had cell respiration and photosynthesis. And now we're going to continue on with cell processes, and we're going to take a look at how cells divide to make new cells. And so just a quick little reminder, the cell theory, there were three parts. We know that the cell is the smallest or basic unit of life, and that every living thing is made up of one or more cells. But one of the parts of the cell theory was talking about where do cells come from, and we know that cells come from pre-existing cells. Now, how do they do that? Well, it's going to be through the process of cell division. And so cell division is just essentially where one cell divides to form two new cells. And we call those new cells daughter cells. And so why would we need for this to happen? Well, we've actually talked about this first one back in the cell unit. And so hopefully you remember that cells are tiny for a reason. The reason most cells have to have a microscope in order to see them is because they are tiny, and they are tiny because we want to maintain a large surface area to volume ratio. And so cells must be tiny to be efficient. Remember, if they get too big, the DNA can get overloaded, the membrane can get overloaded, and the cell just doesn't work properly. And so as cells grow, they're going to get to a point where they're no longer being efficient, and they may get a signal to divide into two in order to be smaller. And so that reduces the volume of the cell. Now we also need cell division to grow and develop. And so, like for instance, you started as one cell. Every person began when sperm met egg and created a zygote, and then that zygote went through rapid cell division. 1 became 2, 2 became 4, 4 became 8. And then as that development continued, these cells became specialized. And so obviously we need that to grow. We also need it for renewal and repair. Like we're constantly having cells die. They are not immortal, and so as cells die, we often replace them, like skin and blood and bone. We also need it to repair. You know, if we had a cut or we had a broken bone or something like that, our body will trigger cell division in order to fix certain tissues. And then just in general, we need it to reproduce. Now, we have two different types of reproduction. We could produce cells that are the same, which is going to be asexual, or we could use it to produce cells that we can use in sexual reproduction. And so when we're talking about unicellular organisms like a prokaryote or an amoeba, essentially when that particular cell goes through cell division, they're producing the whole organism. And one cell becomes two, and we now have two copies of that organism. Now in multicellular organisms, it's a bit more complex. Obviously we have trillions of cells, and so as we mentioned, for organisms like us, it allows us to grow, renew, and repair. And like we mentioned in the last slide as well, the key to asexual is that you have one parent cell and it produces identical daughter cells. And so the key to asexual is that it stays the same. The genetic material is just copied and then split into two new cells. Whereas if it's a sexual process, we're going to get different daughter cells that are unique in their genetic combinations. And then if we're talking about sexual reproduction, this is going to involve cells from two different parents that have to come together. And once again, we're going to get different. Now, an example of asexual reproduction is binary fission, and this is what happens in prokaryotic cells. So remember that prokaryotes are your bacteria and your archaeans. They do not have a nucleus. Their DNA is just free in the nucleoid region. They don't have all of the complex organelles, and so it's a little bit simpler. Essentially, binary fission means to split in half. Binary means two, fission means to split. And so one cell is going to split into two new cells. I don't know all the details, the ins and outs, but essentially the DNA is copied, the cell elongates, the cell membrane divides the cytoplasm, And what you end up with are two identical daughter cells. Okay, so they're going to be just like the parent cell. And this is how our bacteria reproduce. Now the cell cycle is also going to be an asexual process, and the reason it's asexual is because we're getting identical daughter cells. But this is the process that we're going to see in eukaryotes. And this is what we use to make new somatic cells. We're going to see this word pop up again. The prefix soma, S-O-M-A, or some, S-O-M-E, it literally means body. And so somatic cells are all the cells that make up your body. This is going to be skin cells, it could be muscle cells, could be liver cells, but it's just all of the cells that make up your body. And these are the cells that have a full chromosome count. They have 46 chromosomes in a human, and they have all of the genetic information. And so in the process of the cell cycle, we're going to maintain that chromosome number. If we start with 46, our new cells are going to have 46. And that genetic makeup of the cell is going to stay the same. So once again, remember, we're producing identical daughter cells. Now we're going to take a look at these here in a minute. We're going to backtrack and talk about genetic material, but essentially there's kind of three main parts or three important events in the cell cycle. We're going to replicate our genetic material, because if we're going to go from one cell to two cells, we have to make sure that there's genetic material for the new cell. And so it's very important that we replicate our DNA first, and then we're going to divide the nucleus first, and then we're going to divide the rest of the cell, or the cytoplasm. Now, to fully understand the different stages involved over here, you can see that it's a bit more complicated. So in order to understand what's going on in each of these phases, we do need to understand a little bit more about the genetic material in our cells. And so I have two very important words that we're going to see as we go throughout division, and that is chromatin and chromosome. Now, we mentioned this a little bit in our cell unit. We talked about how the chromatin was in the nucleus. And so essentially, both of these are genetic material. Okay, both of these are DNA wrapped around proteins. Those proteins are called histones, and the DNA wraps around them to form these structures called nucleosomes. And the difference between the two is that one is lesser condensed and one is super condensed. Okay, so if we were to look at cells under the microscope, most of the time the DNA is in this form called chromatin, the lesser condensed form. So it is wrapped around proteins, and it's still condensed. We fit about 6 feet of DNA into each nucleus. If we were to stretch it out end to end, you've got a whole bunch of DNA in your nucleus. So it does have to be kind of wadded up a little. This is what I commonly call the noodle form, and I'm going to show you a picture here in a minute and see where I get that term. But it looks like a bowl of noodles, okay, inside the nucleus. And so chromatin is this lesser condensed form. It's what we consider the active form. Because it's lesser condensed, enzymes can get in there and copy it for the sake of DNA replication or for the sake of protein synthesis. Now, most of this time throughout the cell's life, throughout the cell cycle, DNA is going to be in the chromatin form. The other form, the chromosomes, we're only going to see during mitosis, okay, which is going to be division of the nucleus. And what happens is the DNA gets super tightly wound around those proteins, and it forms a very condensed structure called a chromosome. Now, this is what we would consider inactive DNA because it's so tightly wound that those enzymes and proteins can't get in there to copy it. So we only see these during the division stages, and by the time we see it, the DNA has already been replicated. So this is where I get that noodle form terminology. So here's my chromatin inside the nucleus. It super condenses around those histones, and it forms the more compact condensed chromosome. Okay. So as it mentioned in the slide before, it talked about how that the chromosomes, we actually see them during division. And before division occurs, the DNA is replicated. Okay, we'll take a look at the specific stages here in a minute. And so by the time we see the chromosomes, they look like that X that we are familiar with. Okay, So whenever we see a chromosome, typically it looks like an X, and that's because it is a double copy or a duplicated copy. And so when we see this doubled chromosome, it consists of two identical sister chromatids. So this is a sister chromatid on one side, and this is a sister chromatid on the other side. Same thing up here. This is a chromatid, and this is a chromatid. And they're held together at a piece called the centromere. So we have chromatin, the lesser condensed form. During the cell cycle, it gets duplicated, and then when we get ready to divide, it super condenses to form the chromosome. So by the time we see the chromosomes, they are X's, and they consist of two sister chromatids held at the centromere. Now, through the process of cell division, we're going to divide those. And so each chromatid will essentially be its own chromosome in the new cell. Okay, I know that may be kind of confusing. So this next slide, okay, this is a chromosome, all right. This is also a chromosome. The difference is that when you see the X, that means it's a double copy, okay? So same thing up here, chromosome, chromosome, but this one is a duplicated, it's a double copy, okay? So anytime you see that X, just know you have two copies. We don't have two chromosomes, we just have what we call a duplicated chromosome, and that chromosome consists of those two identical sister chromatids held at the centromere. So we have lots of C words there. Now this is just something that you need to be familiar with, and when we get into the next process of meiosis, you'll kind of see this pop up again. But the chromosome number is specific to an organism. So one of the things that makes an organism, a particular type, is the number of chromosomes that it has, and of course the genetic information that's contained within those chromosomes. And I just kind of wanted you to get an idea that just because you have more chromosomes does not necessarily mean that you are more complex. So here we are at 46 chromosomes. Notice that that potato has more than we do, a dog has more than we do, and so the number does not necessarily represent complexity. Okay, you can see that yeast, which is relatively a simple organism, quite a few chromosomes there. Now I did have you put this one in there in your notes. I put it on your note sheet, and that's because when we get to genetics here in a couple of units, the fruit fly is heavily studied, and you can see that it has such a small number of chromosomes, it makes it easier for us to study those genes and where they're located. All right, so now we're going to get into the details of the cell cycle, and this is really what the main meat of our notes today is: the cell cycle and how a cell grows and divides. And so there's two main stages. We can split it up into interphase, which is when the cell is resting and growing and getting ready to divide, and then we have the M phase. Now that can be a little bit deceiving because the M phase are all things division. Okay, this is when the cell is actually dividing, and there's two parts to division. We have mitosis, and that's where that M comes from, but it also includes cytokinesis. So both of these are division processes. Mitosis divides the nucleus, and cytokinesis is going to divide the rest. So all of this is interphase here, and then we have the M phase, which is mitosis and cytokinesis. And so we're going to start off with interphase. Now you don't have to do this, but you do have some space in your notes. You may want to kind of draw a little picture as we go through these various stages. It might not hurt to have a little picture of what that cell would look like in that particular stage. Not a requirement, but just a suggestion. And so in interphase, this is where the cell spends the majority of its life, up to about 90%. And so during interphase, this word actually means between the phases. Inter means between. And so really these are the periods between divisions. Okay, interphase is all of the time between the division stages. And so this is where the cell is being a normal old regular cell. If it's a bone cell, it's being a bone cell. If it's a blood cell, it's being a blood cell. If it's a skin cell, it's being a skin cell. It's doing its job, it's performing all of its functions, but it's also preparing for division. Okay, so this is the majority of a cell's life, and we can divide it into three stages. We have G1, S, and G2. Now, the G's technically stand for the word gap, but you can always associate the G's with the word growth, okay? And then the S phase comes from the word synthesis, all right? So in G1, this is what we consider our first growth phase. The cell is doing its job, it's getting bigger, and it's also doubling the number of organelles that you will find inside of that cell. Now, the S phase is absolutely critical, okay? We have to remember the S phase. It's super, super important because this is where we replicate that DNA. So remember, we're going to go from one cell to two cells, and it's absolutely critical that both cells receive a full copy of genetic instruction. A cell without DNA is useless, okay? And a cell with only half the DNA would be useless. So what we need is our DNA to be replicated so that each cell can have a full copy of genetic instruction. So the S phase is very, very, very important. And then right after the S phase, we have our second growth phase. So the cell is continuing to grow, it's continuing to do normal cell things, and it's also making some proteins and prepping for division. Okay, so all of this is interphase in our cell cycle. Once again, this is up to about 90%. Now, the little small pie piece, I went backwards there, sorry about that. The little small pie piece of the cell cycle is going to be the M phase. We have mitosis, which is nuclear division. And I'm going to tell you right now, this is kind of important, but mitosis is probably the most misused word in biology, okay? A lot of times we substitute the word mitosis for cell division, and that is not really correct, okay? It's very, very common when you're looking at pictures, when you're looking at resources on the internet, when you're watching videos, even me, it's going to be used incorrectly, okay? Oftentimes mitosis is substituted for cell division, but that is not accurate, okay? Mitosis by definition is nuclear division. It is not cell division. Mitosis only divides the nucleus, okay? And mitosis is a term that would only ever relate to eukaryotes, okay? Because if it's nuclear division, prokaryotes cannot do it. So mitosis is specifically a eukaryotic process. So it's okay if you just say mitosis in a wrong way sometimes, as long as you understand that mitosis is not division of the whole cell. Okay, mitosis is just division of the nucleus. This is where we divide those chromosomes that have been doubled, and it involves PMAT. Okay, mitosis is PMAT, PMAT is mitosis. You need to understand that. Mitosis is PMAT, PMAT is mitosis. Now, another stage of division is cytokinesis, and this literally means cyto, cell, kinesis is another term kind of for splitting. And so here we have division of the cell or division of the cytoplasm. And the cytoplasm is all the liquidy stuff and all of the organelles inside. So essentially, it's division of the rest of the cell. And so mitosis is going to give us 2 new nuclei. Cytokinesis will actually give us 2 new cells. So really, it's the cell cycle, it's the combination of mitosis and cytokinesis that is what we would consider cell division. And so now we're going to go through the stages of mitosis, and we're going to take a look at cytokinesis. So remember that mitosis is PMAT, PMAT is mitosis, and so the very first stage is called prophase. Pro means the beginning, and this one is probably the most involved when it comes to mitosis. There's a lot more going on in prophase than in the other stages. Now, prophase is sometimes divided into 2 stages, prophase and prometaphase. So if you ever see that in pictures or on videos, I just want you to know that because there is a lot going on in prophase, in some resources it is split into a prophase and a prometaphase. We are only going to cover it as one, as just PMAT, okay, so just prophase. Now, really we can take all of these bullets here. You can see you've got several bullets, but we can break it down into 3 main events, okay? Number 1, this is where we get the chromosomes. Okay, so this is the point at which that noodly chromatin condenses to form the very compact chromosomes. Now keep in mind, S phase has already happened, so when we see the chromosomes, they look like X's. They have already been doubled, okay? And so chromosomes are the only form of genetic material that can be seen in the light microscope. So this is the point at which they are visible under a microscope. So that's one important component, is this is where we're going to see the chromosomes for the first time, okay? Now, the second main event is we're going to get the spindle, okay? Now, the spindle is just a network of proteins. It comes from the centrosome within the cells. In animals, this involves those additional centrioles, but the spindle is essentially going to be these protein fibers that connect to the chromosomes and pull them apart. There are also going to be some protein fibers that help the cell to elongate and kind of aid in that movement of the chromosomes. So number two, the spindle forms. Now, it's coming from the centrosomes. They're going to attach to the chromosomes. The main thing is this is where we see the spindle. And then the third main event is that the nucleus and the nucleoli are going to go away, okay? Because I can't divide the chromosomes and put them into two new nuclei if I still have one complete nucleus, okay? So even though there's a lot going on here, the three main events that I want you to remember: we get the chromosomes from chromatin, the spindle forms, and the nucleus goes away. Now, the next stage is much, much shorter. Don't forget, you could be drawing these over on the side of your paper. In metaphase, the chromosomes are going to line up in the middle. So you can see that there's not nearly as much going on in this phase, and we do have a little hint in that meta equals middle, meta equals middle, meta equals middle. So the chromosomes line up along the equator. Sometimes that's called the equatorial plate. All it's saying is the chromosomes are lining up in the middle. All right, the next stage, anaphase. Ana equals apart. Now, I don't know that that actually equals apart, but that's how I want you to remember it. And so in anaphase, the sister chromatids are pulled apart. So those centromeres that were holding the chromosomes together are going to be split, or they're going to separate, and then those spindle fibers are going to kind of pull those sister chromatids apart toward opposite sides of the cell, okay? And then our next stage, in telophase, this is where we're going to get two new nuclei, okay? So in telophase, it's kind of the opposite of prophase. So those three things that happened, we went from chromatin to chromosomes in prophase. Well, now we're going to go back to chromatin, the lesser condensed form. We're done with the division. The nuclei are going to reform, so I'm going to see the reformation of my nuclear envelope. And then we're done with the spindle. It's done its job, and so the spindle is going to break down. And so if you look at the pictures over here, okay, I see two new nuclei. Now the problem is I don't have two new cells yet, okay? Remember that mitosis is PMAT. PMAT is mitosis. Telophase is the end of mitosis. And we technically do not have two new cells yet, okay? And so in order to get two new cells, I need cytokinesis. And this is where we're dividing the cytoplasm, which is going to be the rest of the cell. It's going to be the liquidy portion as well as the organelles. And it does look a little bit different in animals and plants. It has to do with the fact that the plants have that cell wall and animals don't. And so in animals, they get what's called a cleavage furrow. It just kind of pinches in. The cell membrane pinches inward until we actually separate the cells or the cell into two brand new ones. And then in plant cells, we get a cell plate. And that's just pretty much a precursor. It's got these little Golgi vesicles, but it's a precursor to the new cell wall that will divide those plant cells. Now this stage technically starts about the same time as telophase. And so in pictures, oftentimes you will see telophase and cytokinesis happening simultaneously. All right, so cytokinesis is not part of mitosis, but it does start happening during that last phase of mitosis known as telophase. So in pictures, a lot of times we're seeing telophase and cytokinesis at the same time. Now, I wanted you to take a look at what this looks like in actual cells, and you're going to do a lab where you look at some of these under the microscope. And so, real cells, they are stained so that we can better see the genetic material. And one thing you have to understand is that this is a continuous process. these cells are fading from interphase into prophase, and then fading from prophase into metaphase. And I'm not sure what's going on with this picture and the spelling of prophase and interphase there. I like the pictures though, that's why I used it. But it's a continuous process. And so a lot of times what we're going to see is like early prophase, late prophase, and then late prophase kind of fades into early metaphase. And then that's going to fade into later metaphase, and then early anaphase, and so forth. So I just want you to understand that there's not just necessarily one look for each phase. What you need to do is just pay attention to what's really going on, okay? So remember that in interphase, I have an intact nucleus. I can see that clear nuclear envelope boundary. I can see that nucleolus intact. In prophase, see how it kind of starts to look broken up and chunky? That chunkiness is because we're getting those chunky chromosomes. And the fact that it's kind of broken looking is because, remember, that nuclear envelope is breaking down. Now you can see by the end of prophase, that nuclear envelope is completely gone, and all we see are those chunky chromosomes. In metaphase, you kind of see this linear pattern. In anaphase, they're kind of starting to move apart. This is like the best look for anaphase. You can see these claws. In telophase, they're kind of more localized, and so we're kind of seeing these two new nuclei. At this point, they're still chromosomes, but they're in the process of going back to chromatin. I can also see that cell plate forming, and so that cytokinesis stage is happening about the same time. See, over here, you couldn't see that cell plate. So that's another indication that you're in telophase and cytokinesis. And then here's what it looks like in an animal cell. So there you see your intact nucleus. You see it's kind of chunky and grainy and broken looking. Here you see that linear pattern. Here you start to see that they're being pulled apart. And then I'm beginning to see two new nuclei and the process of cytokinesis. Okay, so when you're doing this in your lab, those are the kind of the signs that you're looking for. And then the last thing we need to cover is the G0 phase. Now, one thing that you need to understand is that the cell cycle is heavily regulated. In fact, our next lesson is the whole thing is going to be about cell regulation and how we regulate cell division. Okay, this is just a little hint. You got checkpoints and different things that we're looking for. But not all cells are constantly going through interphase, mitosis, cytokinesis, interphase, mitosis, cytokinesis. Not all cells are dividing, okay? And that's because some cells are just too complex, their networks are too complex, the way they're structured and organized in our bodies is too complex, and so we can't just easily divide these cells all the time. So a couple examples of that are muscle cells and nerve cells. They're not constantly going through cell division, and these are both tissues that are not easy to fix. So like if you do have damage to muscle or nerve, they're not easily regenerated. And so what happens is, because some cells, depending on their structures and properties, they don't need to constantly grow and divide, or they can't constantly grow and divide. They actually aren't going through the cell cycle, they exit it and they enter what's called a G0 stage, and this is a non-dividing state. And so literally, if a cell is in G0, they're just being a normal cell. They're not doing the growing and prepping thing that the other cells are doing, and they're not going to constantly divide into two, divide into two, divide into two. So literally, they're just being a normal cell without those cell cycle processes going on. And that is going to conclude our first lesson. As I mentioned, there is a lot of regulation to this, and because there's so much to know, I'm going to create a separate PowerPoint for that. So you guys are now going to spend some time doing some practices, maybe doing a lab, and we're going to quiz over the two cell division notes together. We'll do the cell cycle notes, and then we'll do the cell regulation of the cell cycle, and then we'll quiz over the two of those together. So that's all I have for you today. I hope you guys are doing well, and I will talk to you soon. Goodbye.